Hello everyone, my name is Jeannette Bouchard and I am an infectious diseases pharmacist at WakeMed Health and Hospital System. In this presentation, I will be reviewing the pertinent drug information regarding the SARS-CoV-2 mRNA vaccines.
In this video, we'll be discussing the SARS-CoV-2 vaccines utilizing mRNA technology. There are 24 vaccines utilizing RNA technology in preclinical and 18 in clinical development. One of these vaccines is currently FDA approved in the United States, Comirnaty, which is Pfizer's COVID-19 vaccine and was formerly known as BNT162b2. Another vaccine is currently under EUA approval in the United States, mRNA 1273 from Moderna. These will be the main focus of the video as these mRNA vaccines have EUA or full FDA approval. This video will be updated to reflect data surrounding these the other vaccine candidates as well. Pfizer's BNT162b2 is currently FDA approved as a 2-dose regimen 21 days apart. The storage requirements are -90 to -60 degrees Celsius, and the approximate cost of the vaccine is about $20 per dose. Moderna's mRNA 1273 is currently EUA approved as a 2-dose regimen, 28 days apart, with storage requirements of -20 degrees Celsius and an approximate cost of $32 to $37 per dose.
mRNA technology was first reported in 1990 utilizing reporter gene injections into mice to create proteins. Only a few years later, this technology began to be advocated for as a vaccine platform. Interest in vaccine potential peaked because of mRNA's non-replicative nature, lack of genomic integration, and general instability. These characteristics made it the perfect messenger to carry important information when compared to DNA and viral vector technology. The testing of the first mRNA vaccine was in 1993. The mechanism of mRNA vaccines is depicted in the figure on the slide. RNA encoding the viral spike protein is encapsulated in a lipid coat or lipid nanoparticle. This lipid nanoparticle allows the RNA to enter the cell where it is translated in the ribosome to protein. This protein is then presented on the cell surface to be recognized by CD8 T cells, generating cell-mediated immunity. These protein particles may also be transported outside the cell where they can be identified by other immune cells in the body and create antigen-specific antibodies. There are two classes of mRNAs commonly used as vaccine vectors, non-replicating and self-amplifying mRNA. Both types are currently being studied for prophylactic vaccines for infectious diseases. Concerns with this technology were threefold. 1. mRNA is incredibly unstable and prone to degradation by RNases, which hindered its delivery to cells. Research dealing with delivery systems for mRNA have been a large focus for this technology and involves the use of cationic molecules, lipids, polymers, and nanoparticles. 2. mRNA is highly immune activating since it is recognized by a variety of cell surface and innate immune receptors, a feature of mRNA that could be incredibly beneficial or just as equally detrimental. And 3, it's inefficient in vivo delivery, which has since been improved with the current vaccines by utilizing a lipid-based vector. Other technologies such as microinjections and RNA patches have also been used, but not in the context of the currently studied SARS-CoV-2 vaccines. The first reported use of these lipid-based vectors was in 2015. These lipid-based vectors are internalized and attached to the cell membrane via their non-bilayer lipid phases. As technology has progressed since 1990, many of the original concerns regarding mRNA have been addressed, and more stable and highly translatable products are available and being tested. As of 2020, there were at least 6 phase 1 or 2 clinical trials ongoing for vaccines against infectious diseases such as CMV, Zika virus, and rabies. First, we will review Pfizer-BioNTech's BNT162b1 and b2 candidates. There were 2 primary phase 1 studies for Pfizer's BNT162b vaccines. Notably, during the preclinical and phase 1 data, there were 4 potential Pfizer vaccines, which following clinical trial results in Germany, this 4 was narrowed to 2 potential candidates, which were then tested in the United States. Ultimately, it was decided to only move forward with BNT162b2 for the full phase 3 trial, given the information derived from the phase 1 and 2 data, which will be discussed over the next few slides. BNT162b1 encodes for the receptor binding domain of the SARS-CoV-2 spike protein, and BNT162b2 encodes pre-fusion membrane-anchored SARS-CoV-2 full-length spike protein. During this review, we will focus specifically on the data that compares the two vaccine candidates from Walsh and colleagues, as this directly speaks to the ultimate vaccine candidate, BNT162b2. Groups of participants in the trial were to receive doses of 10, 20, or 30 microgram doses of either vaccine candidate or placebo on a 2-dose schedule 21 days apart. Groups were also broken down by age, with one group including ages 18 to 55 years and the other ages 65 to 85 years. The primary safety endpoints were solicited local reactions and systemic reactions, and the use of antipyretic or pain medication prompted by and recorded in an e-diary. Immunogenicity assessments included SARS-CoV-2 neutralization assay and receptor binding domain or S1 binding IgG immunoassays, performed at baseline, 7 and 21 days after the first dose, and 7 and 14 days after the second dose. Human convalescent sera was included as a benchmark for immunogenicity data. The serologic responses were similar between candidates, and because BNT162b2 is the candidate that fully progresses to phase 3, its neutralizing titers are showcased here. In the figure shown, by day 28, or 7 days after a second dose, in both the younger and older age groups, 30 micrograms of BNT162b2 elicited a neutralizing titer response equal to or above the human convalescent sera. Consistent with the data seen in non-human primates, the highest neutralization titers were seen at 7 and 14 days after the second dose. Of note, for both candidates, there was an overall decrease in antigen-binding IgG neutralization titers seen in the older age group. This effect is similar to what is observed in vaccines for other diseases. The safety data regarding these two candidates is where their differences lie. For BNT162b1, the predominant local reaction reported was pain at injection site, with higher doses eliciting higher local reaction reporting. Local reactions were increased after dose 2 and were reported in similar proportions in both the older, which is not pictured, and younger age groups. Local reactions reported for BNT162b2 were similar to B1. However, there were less reported redness and swelling in the younger age group and no reports in the older age group. There were no grade 4 local reactions reported for either vaccine candidate. The systemic events for BNT162b1 were predominantly fatigue. However, there was a substantial number of participants who reported fever and chills, specifically after dose 2 and in the 30 microgram dose group. Systemic events were less in the older patients when compared to the younger patients. These systemic events peaked by day 2 and resolved by day 7. Systemic events reported for BNT162b2 were lower when compared to B1, as shown on this slide. Following dose 2 of the 30 microgram dose for this candidate, the percent of participants who reported any fever was 17%, compared to 75% reported for the 30 microgram dose of BNT162b1. This pattern was consistent for the older age group as well, which is not shown on this slide. Additionally, after the first dose of BNT162b2, systemic events occurred at similar rates to placebo, especially in the lower dose groups. Based on the data presented in the U.S. phase 1 trial, it was decided to move forward with the best candidate, BNT162b2. This was based mostly on the safety data differences, specifically the systemic reactions, as both candidates produced similar immunogenicity data. Next, we will discuss the pivotal efficacy trial for BNT162b2. CS4591001 is an ongoing phase 3 placebo-controlled, observer-blinded efficacy trial for participants ages 16 and older, an amendment that was added in September for the U.S. specifically. This trial evaluates the current data on safety, immunogenicity, and efficacy of BNT162b2. Data regarding durability of immune responses and immunogenicity is ongoing and not included in the published dataset. Participants included were healthy or had stable chronic medical conditions, including HIV, hepatitis B, and hepatitis C. Patients were excluded if they had a medical history of COVID-19, were immunocompromised, or were on immunosuppressive therapy, were pregnant, or were breastfeeding. The primary efficacy endpoints were confirmed COVID-19, defined as a positive SARS-CoV-2 nucleic acid amplification-based test with one or more mild COVID-19 symptom, with onset greater than or equal to 7 days after the second dose, in patients without serologic evidence of SARS-CoV-2 at randomization. A second primary endpoint included this outcome in patients both with and without serologic SARS-CoV-2 evidence at randomization. Secondary efficacy endpoints included efficacy against severe COVID-19, defined as having serologic evidence of SARS-CoV-2 with clinical signs indicative of severe systemic illness such as respiratory failure, ICU admission, or death. The primary safety endpoints of the trial were solicited local and systemic adverse events, the use of antipyretic pain medications, and unsolicited adverse events. 44,820 participants were screened and 43,548 were randomized at 152 sites worldwide, including the United States, Argentina, Brazil, South Africa, Germany, and Turkey. 43,448 participants received injections and 37,706 had 2 months of available data as of October 9th. The baseline characteristics for this group are shown here. 50.6% were male, 82.9% white, with the majority, about 57.8%, falling into the younger age group. Notably, the participants ages 16 to 17 had a limited representation of 153 at the time of this report. Despite efforts to ensure diversity by enrolling participants on a global scale, 76.7% were enrolled in the United States, with Argentina having the second most participants enrolled at 15.3%. 35% of participants had a BMI greater than or equal to 30, and 20.5% had a baseline comorbid condition classified according to the Charlson Comorbidity Index. Of note, HIV patients were included in the safety population. However, their safety data did not contribute to the overall analysis of this population at the time. Overall baseline characteristics in the 2-month follow-up population were similar between treatment and placebo arms. Of the approximate 36,000 participants who had no evidence of infection prior to enrollment, there were 8 in the treatment arm and 162 in the placebo arm who were observed to have symptomatic COVID-19 at least 7 days after the second dose. This equates to a vaccine efficacy of 95%. For the secondary primary efficacy endpoint, in those patients with and without prior infection at enrollment, there were 9 and 169 cases in the treatment and placebo arms respectively, which equates to a 94.6% vaccine efficacy, demonstrating that the true vaccine efficacy in the population most consistent with the real-world scenario is at least 89.2% with the current data. For the secondary efficacy endpoint of prevention of severe COVID-19 disease, there was 1 case reported after 1 dose in the treatment arm and 9 in the placebo arm in the modified intention to treat population, which equates to a vaccine efficacy of 88.9%. When looking at severe COVID-19 cases occurring at least 7 days after dose 2, there was 1 case in the treatment arm and 4 in the placebo arm, which equated to a vaccine efficacy of about 75%. Due to the small number of severe COVID-19 cases available, the posterior probability that the vaccine efficacy for this endpoint was greater than 30% did not meet the pre-specified criteria of greater than 98.6%, and these results should be interpreted cautiously until more data is available. The efficacy between the first and second doses was 52%, which suggests that there may be some protection following the first dose, but for optimal protection, both doses need to be administered. This study was not designed to evaluate the efficacy of following 1 dose, and thus the follow-up time frame for this data is short. When subgroups were analyzed, outcomes were generally consistent with the overall population, including those with hypertension and in high-risk groups such as older age and obesity. Although it is important to note that this study was not powered to assess efficacy in these subgroups. When looking at the local reactogenicity population, there were more local reactions in the treatment group than the placebo group. However, most were mild to moderate in nature. Severe local reactions had a frequency of less than or equal to 0.6%, with no grade 4 or potentially life-threatening reactions reported. Injection site pain was the most frequently reported local symptom, and this was also reported more frequently in the younger age group than the older age group, as shown in the graphs. The younger population is located at the top, and the older population is the bottom. Overall, local reactions were similar after dose 1 when compared to dose 2, with redness and swelling being the most consistent. Most reactions resolved within a few days of onset and were largely driven by injection site pain. Systemic reactogenicity outcomes again were higher in the treatment group than in the placebo group, with the exception of vomiting and diarrhea, which was similar to placebo. Certain reactions had increased frequency after dose 2, such as fever and muscle pain. Overall, the most noted systemic reactions were fatigue and headache. Again, older patients seemed to be less likely to have systemic reactions than younger patients. After both doses, the use of antipyretic or pain medication was slightly lower in the older group compared to the younger group, and trending similarly with fever, which increased after dose 2. Most of the systemic events were mild to moderate in severity. Severe systemic reactions were rare and occurred in less than 2% of patients. It is important to note that systemic reactions were observed within a median onset of 1 to 2 days following vaccination and resolved within a median duration of 1 day for both younger and older age groups. For overall adverse events, as mentioned earlier, there were data available for 2 months post-dose 2 in 37,706 participants, but results for the total enrolled population are shown here. There was an increased adverse event rate in the BNT162b2 arm versus the placebo. However, when looking at serious adverse events or events leading to study withdrawal, BNT162b2 was comparable with the placebo, with rates being less than 1%. There were no clinically meaningful differences in adverse events when broken down by subgroups of age, sex, race, or baseline SARS-CoV-2 status. When broken down by the group that had 2 months of follow-up following dose 2, rates of adverse and serious adverse events were similar. There were 64 reports of lymphadenopathy in the treatment arm compared to 6 in the placebo arm, with the majority of these being considered related to study intervention. These were reported within 2 to 4 days of vaccination and continued for an average duration of 10 days. Of the 6 reported deaths in the study, 2 in the BNT162b2 arm and 4 in the placebo arm. None of them were related to vaccine, placebo, or COVID-19. It is important to note that there were 4 reports of Bell's palsy in the BNT162b2 arm and 0 in the placebo. Although these rates are consistent with the expected background rate, it is an area noted to be watched closely in the future due to the numerical imbalance between groups. 1 case of Bell's palsy was noted to have resolved at the time of data cutoff, and the other 3 were reported as continuing to resolve.
6-month data for BNT162b2 safety and efficacy up until March 13, 2021 was made available in July. In this update to the Phase 3 clinical trial, 42,094 patients without prior SARS-CoV-2 infection were evaluable in both the blinded and open-label periods of the study. This data is reflective of patients greater than or equal to 12 years old. However, the majority of participants were greater than or equal to 16 years old. For those patients that are evaluable, vaccine efficacy greater than or equal to 7 days after the second dose corresponded to 91.3%, with a 95% confidence interval of 89 to 93.2. Vaccine efficacy against severe COVID-19 after dose 1, based on the FDA definition, had a point estimate of 95.7% with a 95% confidence interval of 73.9 to 99.9. Of note, there were numerically similar number of deaths between groups and the causes of death were balanced. These data are encouraging as we continue to follow the long-term safety and efficacy outcomes for these vaccines. Asymptomatic infection was not assessed in the landmark trial that allowed for BNT162b2 to be EUA approved. Real-world effectiveness has been relied on for data surrounding BNT162b2 in asymptomatic infections. A study out of Israel analyzed positive SARS-CoV-2 tests following 1 dose of BNT162b2 vaccine. In this study, they found that the viral load is reduced 4-fold for infections occurring 12 to 28 days after the first dose of vaccine. These findings show the potential benefits of lowering infectiousness and viral shedding earlier in immunization series. Some limitations of this study were its matched controlled design, as they were not able to control for variables including differences in variants, behaviors, and tendencies to be tested. Another limitation was that they used oronasopharyngeal tests, which are not the best when correlating to viral shedding and infectiousness. A second study out of the UK also assessed the effect of one dose of BNT162b2 on test positivity rates and cycle threshold values in asymptomatic patients. Patients were tested weekly in the UK program to assess for asymptomatic infection. There were 4 out of 1,989 positive tests greater than 12 days after the first dose, compared to 26 out of 3,252 positive tests in unvaccinated patients, representing a 4-fold decrease in a risk of asymptomatic SARS-CoV-2 infection. Limitations to this study are similar to the Israel data. Notably, both of these studies were conducted following the first dose of BNT162b2, suggesting early reduction in asymptomatic infection. More data in observational retrospective studies have continued to be published as shown on the slide. Asymptomatic infection data are difficult to compare across studies as the study designs are often different in the way vaccine effectiveness is calculated and how asymptomatic infections are found and defined. Because of these differences, it's important to interpret this data with caution. Overall, thus far, the data suggests that vaccines do provide benefit for asymptomatic infection, especially with our earlier variants in the pandemic. These results shown are for BNT162b2 study with adolescents aged 12 through 15 years. These data include safety and efficacy data for up to 2,260 participants, with 1,308 participants followed for greater than 2 months after the second dose of vaccine. The cutoff point for these data was March 13, 2021. Demographics were similar at baseline, with 49% of patients being female and 85.9% of patients being white. Both local and systemic reactogenicity was assessed. Local symptoms after dose 1 lasted for around 2 days, with higher rates for both local and systemic reactions being higher in the treatment arm compared to placebo. Systemic reactions were slightly higher following dose 2 of the series. Serious adverse events were reported in 0.4% of participants in the treatment arm compared to 0.1% in placebo. The only numerical imbalance seen was in non-serious adverse events, where there were numerically more participants reporting lymphadenopathy in the treatment arm. These were considered to be plausibly related to the treatment arm. Vaccine efficacy 7 days after dose 2 of the series for adolescents was 100%, with a 95% confidence interval of 75 to 100% for participants without prior SARS-CoV-2 infection. When including participants with prior SARS-CoV-2 infection, vaccine efficacy was very similar, although this was a small portion of patients. There was an additional immunogenicity study completed on a subset of randomly selected participants that demonstrated non-inferior immune response compared to participants who were 16 to 25 years old, who had no serologic evidence of SARS-CoV-2 at baseline. The time point for the studies was 1 month after dose 2. These data demonstrate that the 2-dose BNT162b2 vaccine series is both safe and effective in adolescents aged 12 to 15 years. Data are emerging to try and characterize the breakthrough infections post-vaccination and the immune correlates for them. Currently, one study out of Israel sought to identify breakthrough infections in healthcare workers via extensive evaluations of those who were symptomatic or had known exposures to COVID. Correlates of breakthrough infection were assessed in a case control analysis where patients were matched with 4 to 5 uninfected controls who had antibody titers obtained a week before SARS-CoV-2. The majority of the 39 breakthrough infections were due to B117. The predicted GMT of peri-infection neutralizing antibody titers was 192.8, with a 95% confidence interval of 67 to 549 for cases, and 533 with a 95% confidence interval of 408 to 698 for controls, for a predicted case-to-control ratio of neutralizing antibody titers of 0.361. It's important to note the limitations of this study. 1. Breakthrough infections were predominantly in young, unhospitalized patients, with 27 infections found solely because of exposure to a person with known SARS-CoV-2 infection. Many were asymptomatic, so around 33% during the duration of infection. Additionally, the matched controls were only matched based on the availability of antibodies and not on testing and exposure, and therefore it is difficult to truly compare to the case population. Immune correlates of protection will be important in the future for driving whether antibody decay affects clinical outcomes. In summary, based on current data from both preclinical and clinical trial data, BNT162b2 is safe and effective against COVID-19 with 95% vaccine efficacy in adolescents down to 12 years old. Serious adverse events were low and consistent between treatment and placebo arms, including in patients who were SARS-CoV-2 positive and asymptomatic at baseline. It's important to note that the systemic side effects were more frequent in the treatment arm, and this should be taken into consideration when vaccinating groups of people such as healthcare workers or counseling your patients prior to vaccination. The current data for BNT162b2 provides proof of concept for RNA-based protection against infectious diseases, which will be huge for the future of this field. The downfalls of this vaccine series continue to be centered around cold storage requirements, as this limits its utility across the globe. Data will hopefully continue to fill the gaps for immunosuppressed patients, patients who are pregnant, and efficacy against variants. Notably, available data for patients who are immunosuppressed, pregnant, and SARS-CoV-2 variants are presented at the end of the video in combination with mRNA data that is available. Upcoming data for BNT162b2 includes trials for pregnant women, children under 12 years old, effects of a booster dose on immunogenicity, especially in the setting of the newfound variants, and data on a lyophilized formulation that will allow for more stable storage. Next, we will review the available data for Moderna's RNA-based vaccine candidate mRNA-1273. The Phase 1 preliminary report for mRNA-1273 was published on July 14, 2020, with additional data on older adults being published originally in September. This was a dose escalation open-label trial of 45 adults 55 years of age or younger and 40 adults 56 years and older. Participants received 2 vaccinations separated by 28 days of 25, 100, or 250 microgram doses. Due to increased reactogenicity without significant immunogenicity, the older age group only received the 25 microgram and 100 microgram doses. Follow-up visits were on days 7 and 14 following each vaccination and on days 57, 119, 209, and 394. Of note, participants were asked to not use antipyretic or pain medications before or after vaccination. Immunogenicity data from this study indicated that a 100 microgram dose as 2 injections 28 days apart resulted in neutralizing antibody induction by day 7 post-dose 2, as seen on this slide. Specifically, in the 100 microgram and 250 microgram dosing groups, by day 7 after dose 2, geometric mean titers for neutralizing titers were similar in magnitude to each other and were comparable to the upper half of the convalescent sera distribution. Again, similar to preclinical data, T-cell responses strongly favored the Th1 T-cell expression, and there were low-level Th2 T-cell expression. In participants who were 56 years or older, similar high-level neutralizing and IgG antibody binding was exhibited to the younger participant group. As shown on the slide, neutralization increased rapidly 7 days after the second vaccination, further emphasizing the importance of the second vaccine in both age groups. This data is based on a smaller number of participants than included in the younger age group, and full immunogenicity data will likely need further elucidation in this older population. Next, we will discuss the pivotal efficacy data for mRNA-1273. Study 301 is an ongoing Phase 3 placebo-controlled efficacy trial for participants aged 18 and older. This trial evaluates the current data on safety, immunogenicity, and efficacy of mRNA-1273. Of note, this data is from the available data that was last updated on November 25, 2020 from Moderna's EUA approval briefing document. Participants included were healthy or had stable chronic medical conditions such as HIV, hepatitis B, and hepatitis C. Patients were excluded if they had a medical history of COVID-19, were immunocompromised or on immunosuppressive therapy, were pregnant, or were breastfeeding. They were randomized in a 1-to-1 ratio that was stratified based on age and presence or absence of risk factors for severe illness of COVID-19. Treatment groups included 2 100 microgram doses of mRNA-1273 28 days apart and 2 doses of placebo 28 days apart. The primary efficacy endpoints were confirmed COVID-19 that was defined as a positive SARS-CoV-2 test with 2 systemic or 1 respiratory COVID-19 symptom, with onset greater than or equal to 14 days after the second dose in patients without serologic SARS-CoV-2 at randomization.
Secondary efficacy endpoints included efficacy against severe COVID-19, which was defined as having SARS-CoV-2 with clinical signs indicative of severe systemic illness such as respiratory failure, ICU admission, or death, COVID-19 regardless of symptomatology or severity starting 14 days after the second injection, and symptomatic COVID-19 based on a less restrictive criteria of one or more symptoms. The primary safety endpoints of the trial were solicited local and systemic adverse events, the use of antipyretic pain medications, and unsolicited adverse events. All participants assessed provided a nasopharyngeal swab and blood sample for SARS-CoV-2 before the first and second injection during the study. 3,381 participants were randomized. Overall, 52.6% were male and 79% white, with a majority of participants being between the ages of 18 and 65. The study had planned to enroll at least 25% and up to 50% of participants most at risk for severe complications, which based on the full analysis report on November 25th was achieved, with 41% being above 65 years old or younger than 65 with a severe risk factor such as diabetes, severe obesity, or chronic pulmonary disease. As with the clinical trials for BNT162b2, participants included were at high risk for disease acquisition based on their occupation. In Study 301, about 25% of participants were healthcare workers, with the remainder being other frontline workers that would put them at risk of disease acquisition. Baseline characteristics between groups appear to have been well balanced between treatment and placebo arms based on the available data. For the primary endpoint of symptomatic COVID-19, at least 14 days following the second injection in baseline seronegative patients, there were 11 cases in the treatment arm and 185 cases in the placebo arm, correlating to a vaccine efficacy of 94.1%. The slide shows the interim dataset cumulative incidence rate of time to first occurrence of COVID-19 starting 14 days after the second injection as of November 25th. With the red line representing mRNA 1273 and the blue line representing placebo. For symptomatic COVID-19, regardless of prior SARS-CoV-2 infection, there were 12 cases in the treatment arm and 187 cases in the placebo arm, with a vaccine efficacy of 93.6%, which is consistent with the primary outcome, but is more representative of the real-world population. Since only one participant who was previously COVID-19 positive had symptomatic COVID-19 disease, these results should be cautiously interpreted and further evaluation is needed. Regarding secondary endpoints, there were 0 cases of severe COVID-19 in the treatment arm and 30 in the placebo arm, correlating to a point estimate of vaccine efficacy of 100% for preventing severe disease. One of the cases of severe COVID-19 disease unfortunately resulted in death in the placebo arm. In patients who had only received 1 dose of vaccine at the November 11th interim analysis cutoff, vaccine efficacy was 80.2% with a median follow-up time of 28 days. With such a short follow-up period in a trial that was not designed to assess this vaccine after 1 dose, there is not sufficient data to suggest that 1 dose is appropriate and a full 2-dose schedule is needed for optimal protection. In the modified intent-to-treat population, which included participants who were in the full analysis population who had no immunologic or virologic evidence of COVID-19 on day 1 before the first dose, when evaluating all definitions of COVID-19, the conservative COVID-19 definition of the primary efficacy endpoint, secondary definition of COVID-19, and those who were PCR positive at pre-dose 2 scheduled visit, the vaccine efficacy was 89.5%. Notably, the number of asymptomatic cases between doses 1 and 2 were numerically lower in the mRNA 1273 group, showing an overall 62% reduction in asymptomatic infection. This finding is limited by the fact that it only represents 1 dose of a 2-dose series and a short follow-up period after that first dose. More data are needed to ascertain the true benefit of this vaccine in protecting against asymptomatic disease. When looking at the local reactogenicity population, there were more local reactions in the treatment group than the placebo group. However, most were grade 1 to 2 in severity. The most common solicited reaction was pain, and the incidence was comparable between doses. Grade 3 local reactions were more common after the second dose, and the driver of this was injection site pain. The majority of reactions occurred on the day of vaccination or the day after and generally persisted for about 3 days. There were a few local reactions that persisted beyond 7 days in the treatment arm, but not in the placebo arm, and these were mainly induration, erythema, and pruritus. In a subgroup analysis of adults greater than or equal to 65 years of age or older, rates of solicited reactions and other unsolicited adverse events were comparable to all other participants. Systemic reactogenicity outcomes again were higher in the treatment group than in placebo. Overall, solicited systemic events increased after dose 2, with about 55% reporting after dose 1 and about 79% reporting following dose 2. Overall, the most noted systemic reactions were headache, fatigue, and muscle pain. Most of the systemic events were grade 1 or 2 in severity. The most common grade 3 reactions occurred after the second injection and were fatigue, myalgia, headache, and arthralgia. It is important to note that systemic reactions were observed within a median onset of 2.6 days after the first dose and 3.1 days after the second dose, and resolved within a median duration of 1 day for both the younger and older age groups. Solicited adverse events were less common in participants who were positive for SARS-CoV-2 at baseline when compared to those who were negative at baseline. For overall adverse events, data was available for 28 days post any vaccination in 30,351 participants and are shown here. Unsolicited severe and serious adverse events were similar between treatment arm and placebo. The most commonly reported unsolicited adverse event after injection was headache. Serious adverse events, which were defined as adverse events that prevented the participant's daily activity, that had a greater frequency in the treatment arm regardless of age group, included those related to local and general reactogenicity reactions, such as injection site pain and headache. There were 13 deaths as of December 3rd, 6 in the treatment arm and 7 in placebo. None were considered to be related to vaccine or placebo, and only 1 was confirmed to be related to COVID-19, which was in the placebo arm. There were no anaphylactic or severe hypersensitivity reactions noted at the interim analysis, and there were 4 total reports of Bell's palsy, 3 in the treatment arm and 1 in the placebo arm. Currently, there are insufficient information to determine a causal relationship between reports of Bell's palsy and vaccination. Interim results from the phase 1 trial describes mRNA 1273 elicited antibodies in 33 participants stratified according to age at 180 days after the second dose of a 100 microgram dose of vaccine. Antibody activity for receptor binding domain and pseudovirus neutralization assay were high in all age groups at day 209. The estimated half-life binding of antibodies after day 43 was 52 days using the exponential decay model, which assumes a steady decay rate over time, or 109 days, which was calculated with the use of the power law model, which assumes decay rates decrease over time. For neutralization antibody half-life, it was 69 and 173 days with each model, respectively. These data demonstrate that antibodies elicited by mRNA 1273 persisted for 6 months following dose 2, and although we do not have the best correlate of vaccine efficacy, these data are encouraging.
These results shown are for mRNA 1273's study with adolescents aged 12 through 17 years. These data include safety and efficacy data for up to 2,489 participants who received vaccine with a median follow-up duration of 53 days after the second dose. Demographics were similar at baseline, with 49% and 84% of patients being female and white, respectively. Both local and systemic reactogenicity was assessed. Local symptoms and systemic reactions lasted generally about 4 days, with higher rates for both local and systemic reactions in the treatment arm compared to placebo. Systemic reactions were slightly higher following dose 2 of the series. Local reactions that persisted beyond 7 days were higher in the mRNA 1273 group after the first injection. These were primarily axillary swelling or tenderness. There were also higher local reactions with an onset of 7 days in the mRNA 1273 group, and these were again axillary swelling, tenderness, and erythema. There were no deaths, MIS-C, myocarditis, or pericarditis observed at the time of publication. Vaccine efficacy 14 days after dose 2 of series for the adolescents was 100% for participants without prior SARS-CoV-2 infection. This should be interpreted with caution, as there were no cases seen in the mRNA group and only 4 cases in the placebo group. For patients with asymptomatic SARS-CoV-2, 14 days after dose 2 in the modified intention to treat population, the point estimate for vaccine efficacy was about 60%. There was an additional immunogenicity study completed on a subset of randomly selected participants that demonstrated non-inferior immune response compared to participants who were 18 to 25 years old who had no serological evidence of SARS-CoV-2 at baseline. The time point for these studies was 1 month following dose 2. These data demonstrate the 2-dose mRNA 1273 vaccine series is both safe and effective in adolescents aged 12 to 17 years. Limitations of this study are that the efficacy endpoints were secondary objectives due to the nature of the disease. Adolescents often have milder COVID-19 disease and lower disease incidence when compared to adults. Given this, more robust data may be needed to truly generate an assessment of the vaccine efficacy for mRNA 1273 in this population. However, the current data looks great. Data are emerging to try and characterize the breakthrough infections post-vaccination and the immune correlates for them. Currently, there are data from the COVE trial, which is the phase 3 trial for mRNA 1273 that assesses 3 different SARS-CoV-2 antibody markers. The figure here shows the covariate adjusted cumulative incidence of COVID-19 by low, medium, high tertile of day 57 pseudovirus neutralization titer. The gray line represents placebo, the solid line considered low titer, dashed line medium titer, and dotted line was high titer. Authors found that all 4 markers assessed, the IgG binding antibodies to spike and the spike receptor binding domain, as well as ID50 and ID80 neutralizing antibody titers, were inverse correlates of risk through 4 months post-dose 2. Interestingly, there was a positive vaccine efficacy for recipients who were undetectable, indicating that vaccine efficacy is not fully mediated through antibody markers and other aspects of the immune system are at play post-vaccination. It is important to note the limitations of this study. 1. Correlates for COVID-19 outcomes and severity of disease were not assessed, thus making it difficult to ascertain if infections were mild in nature or more severe, which would be a greater concern. 2. Infections were from earlier in the pandemic, and since then we have had several variants emerge. Although data that is discussed later in the presentation suggests vaccine efficacy continues with these variants. Last, there was a low number of participants with undetectable/low titers, making it difficult to truly establish a correlate of protection with these data. Immune correlates of protection will be important in the future for driving whether antibody decay affects clinical outcomes. In summary, based on current data from both preclinical and clinical trial data, mRNA 1273 is safe and effective against COVID-19. Serious adverse events were low and consistent between treatment and placebo arms, including in patients who are SARS-CoV-2 serologic positive and asymptomatic at baseline. Again, just as with BNT162b2, it's important to note that the systemic side effects were more frequent in the treatment arm, and this should be taken into consideration when vaccinating groups of people or counseling patients on vaccination. The current data for mRNA 1273 provides proof of concept for RNA-based protection against infectious diseases, which is huge for the future of this field. The downfalls of this vaccine series continue to be centered around cold storage requirements, as this limits its utility across the globe. Data will hopefully continue to fill gaps for patients who are immunocompromised and those who are pregnant. Future studies involving mRNA 1273 to help fill gaps in knowledge include those involving heme-onc populations, adolescents and children, transplant patients, and to study the effect of a booster dose on immunogenicity, with the booster specifically being designed to target variant strains of the virus. Both Pfizer and Moderna's vaccine candidates were approved for emergency use authorization earlier in the pandemic, and as of August 23, 2021, Pfizer's vaccine candidate Comirnaty is fully FDA approved for ages 16 and older. There are a few key differences between these two vaccines that are important to know for healthcare workers and vaccine recipients. The Pfizer-BioNTech vaccine is 2 doses, 21 days apart, for ages greater than or equal to 12 years old for EUA and 16 and older for full approval. It also must be stored at -80 degrees. There are about 5 doses in the vial, although there are reports of many being able to have enough left for a 6th dose and a sporadic 7th. This vaccine also is not interchangeable with other vaccines such as Moderna at this point in time. For Moderna's vaccine, it requires 2 doses 28 days apart and is approved for ages 18 and older. It should be stored at -25 degrees Celsius and is good for up to 12 hours once punctured. There are 10 available doses per vial, and it has no preservatives and is not interchangeable with other vaccines at this point in time. Moderna's vaccine is currently under European Medicines Agency evaluation for conditional marketing authorization.
Other pertinent severe side effects between the two vaccines are rates of Bell's palsy and anaphylaxis. The rates of Bell's palsy during both Moderna's and Pfizer's clinical trials are consistent with background rates in the population. However, given the numerical imbalance between treatment and placebo arms, this event is also being followed closely during emergency use. During ACIP's January 27, 2021 meeting, updates regarding anaphylaxis reported to the VAERS system were given. There have been a total of 71 cases from December 14th to January 18th. 50 cases followed Pfizer vaccines and 21 cases followed Moderna's. Anaphylaxis following vaccines is a rare occurrence, typically at a rate of 1 per million doses for most vaccines, according to the CDC. Currently, Pfizer's vaccine has a rate of 5 per million and Moderna 2.8 per million. Most of these cases of anaphylaxis were within 30 minutes of injection. There were a handful of outliers, with one case occurring 20 hours after injection. Most patients had a documented history of allergies or allergic reactions to food, with about 24% having a history of prior anaphylaxis.
Recently, with the rollout of pediatrics, myocarditis came to the forefront as several cases were reported to the VAERS system following Pfizer's vaccine. The Vaccine Safety Technical Work Group at the CDC stated a higher number of observed than expected myocarditis or pericarditis cases in 16 to 24-year-olds, and further analysis are needed. Initially, there were 475 cases of myocarditis following vaccination in people under 30 years old, with 81% having full recovery of symptoms and 19% having ongoing symptoms or of unknown status. Israel has also reported cases of myocarditis. They have been vaccinating 12 and up since the beginning of June and started vaccinating 16 and up in January. Their statement is that there appears to be a probable link in men aged 16 to 30, but the benefits outweigh the risks, and they continue to vaccinate. Since these reports, there have been a number of retrospective studies and case series regarding myocarditis following vaccination with mRNA vaccine candidates. Across most reports, the populations affected continue to be males aged 16 to 24 with resolution of symptoms occurring shortly after hospitalization. Short-term symptoms of this ADE seem to be mild and resolve. Long-term effects are not yet known as longer follow-up are needed. On June 18th, the ACIP advisory meeting determined that the benefits of mRNA COVID-19 vaccines clearly outweigh the risks in all populations, including adolescents and young adults. Given pregnancy and breastfeeding was an exclusion in the initial clinical trials for both mRNA candidates in the United States, real-world data has been important to determining safety and efficacy in these patients, as well as safety and efficacy for the baby. In a V-Safe database pulled from a total of 35,691 people who were identified as pregnant, there were no obvious signs or safety signals in pregnant patients. These data are encouraging, and V-Safe and VAERS data are continuing to be evaluated as more people are vaccinated with these novel vaccines. 2 studies evaluated immunogenicity in patients who are pregnant and breastfeeding anywhere from 2 to 8 weeks after the second dose of mRNA vaccine. In the study by Gray and colleagues, which included 84 patients who were pregnant and 31 who were lactating, titers for vaccine-induced antibodies were equivalent to non-pregnant women. The titers included were of SARS-CoV-2 spike and RBD, IgG, IgA, and IgM. These were quantified in sera and breast milk at baseline, at second vaccine dose, 2 to 6 weeks post-second vaccine, and at delivery. Notably, in this study, neutralizing antibody titers were numerically lower in umbilical cord than in maternal sera, but this difference was not significant. These data support the current recommendations by ACOG and the CDC for patients who are pregnant and or lactating in regards to the mRNA COVID-19 vaccines. Genetic variants of SARS-CoV-2 have been circulating across the world throughout the pandemic. Some key variants that have led to assessment of vaccine effectiveness are the Alpha, Beta, and Delta variants. As of September 2021, in the United States, the Delta variant makes up about 98% of the cases for COVID-19. Given this abundance, it is important to assess whether the mRNA vaccines continue to be effective against Delta. On the slide, I have highlighted a number of observational studies across the globe. Across the top of the graph, you can see the varying countries these observational studies were conducted in, with the vaccine efficacy on the y-axis. The different colors represent the vaccine efficacy against different types of SARS-CoV-2 infection. Patients who are just simply defined as infection, patients who are defined as symptomatic, hospitalized, and death. There have been a number of these observational studies. Most of them suggest that vaccine protection against severe disease, hospitalization, and death remains high. These are the darker colors on the slide, which is greater than 80% in most cases. When looking at vaccine effectiveness against infection, there does seem to be a mild decrease in Delta vaccine. However, these observational studies need to be cautiously interpreted and compared. Some factors that may cause pause when looking at these data are differences in the study methods, differences in dosing intervals, specifically for the UK and Canada, which have longer intervals than the US and Israel, the timing of vaccination for certain populations. In most countries, older groups were vaccinated first, which was earlier in the pandemic, and a lifted mask mandate and lockdowns, which coincidentally happened at the same time as Delta took its surge in most countries. Overall, the most important point to take away from these observational data is that the Delta vaccine effectiveness for severe disease continues to be excellent. And these vaccines work against all current and past circulating SARS-CoV-2 variants. On August 12, 2021, the FDA updated the EUA for both Pfizer and Moderna's vaccine to allow for an additional dose in patients who are moderate to severely immunocompromised. This additional dose should take place 28 days following the second dose of either vaccine candidate. ACIP specifically outlines who this additional dose would be approved for, which includes patients who are being treated for solid tumor or heme malignancies, solid organ transplant recipients on active immunosuppressive therapy, and advanced or untreated patients with HIV. This additional dose is not a booster. There is a difference. An additional dose is needed when the initial immune response following the primary series is likely insufficient, meaning there were no antibodies or few antibodies created during the initial doses. An additional dose would be used to assist with seroconversion in certain patient populations. A booster is for when the primary vaccine series is likely to have waned over time. This is specific for patients in the situation where they have seroconverted, but over time the clinical effectiveness may be diminished and an additional dose is needed to continue the protection, similar to our tetanus boosters. The additional dose for patients who have received Pfizer or Moderna stems from available data on antibody levels in immunocompromised patients. Most notably was a study from Hall and colleagues that was a double-blind, randomized controlled trial of a third dose of mRNA 1273 at 2 months or 2 months following the second vaccine dose in the series. They enrolled 120 organ transplant patients without a previous COVID-19 diagnosis. The median age was 66 years, and the median time from transplant to third dose was about 3 years. After the third dose, the percent of patients above the 30% threshold for neutralizing antibody positivity was 60% in the mRNA 1273 group and 25% in the placebo group. Notably, local and systemic reactions were more common after the third dose when compared to placebo, but there were no grade 3 or 4 events and no cases of acute rejection. One major strength of the study was that it had a placebo arm and was able to better showcase the improvement in antibody levels following the additional dose. A second study by Kumar and colleagues looked at giving 101 patients a third dose 2 months following the second dose, and they found that of the 59 patients who had not seroconverted prior to the third dose, 26 or 44% had seroconverted by 4 weeks following the third dose. These data support the use for an additional mRNA vaccine dose for those who are moderate to severely compromised. However, the clinical data for this additional dose is lacking.
Specific resources to learn more about vaccine recommendations and frequently asked questions are listed here on this slide. The CDC provides excellent guidance for both healthcare providers and patients receiving vaccine, as well as separate information for both EUA-approved vaccines available. Thank you very much for watching. I hope this was a helpful summary of the SARS-CoV-2 mRNA vaccines. Again, this video will continue to be updated as information becomes available. If you have any questions regarding the material presented in this video, please reach out.